All right, my name is Meg Graham, and today I'm going to talk to you about erythrocyte physiology and erythropoiesis. But before we can start doing that, we really need to go back and talk about hematopoiesis. And you say, well, what's hematopoiesis? Well, that's basically the production of the formed elements of the blood. And you're going, well, what's the formed elements of the blood? Well, that's what you and I would call cells, blood cells. Now, we don't technically refer to them all as cells because they're not truly all cells, and we'll talk about that in just a minute. But anyway, this hematopoiesis, or this production of blood cells, occurs in red bone marrow. In the axial skeleton, you're going to find it occurring in the bones of the cranial bones, the flat bones, the bones of the vertebra. Then you have it occurring in your girdles, like in your sternum, your clavicle, and then in the pelvic girdle, you're going to have it in the ossa coxa. And then also you're going to have this taking place in the humerus and femur in the epiphyses of these bones. We've got a constant turnover of blood cells, and this is because you have a very short lifespan in these blood cells. As you'll see, a lot of them don't have nuclei, and so they don't have anything to keep them going. So we have to constantly be replacing them and destroying them and replacing them and destroying them and so forth. Well, if you're going to be replacing a lot of cells, then you've got to have a stem cell population. And the stem cell for all the blood cells is called the hemocytoblast. And that's going to help us make our red blood cells, our white blood cells, and all the different formed elements of the blood. Well, let's look first at the leukocytes, or the white blood cells. Now, this is going to include things like the neutrophils, eosinophils, basophils, lymphocytes, and monocytes. And these are your only true blood cells. And the reason they're the only true ones, if you look at all these are going to be these larger cells here. They all have nuclei, and the nucleus keeps the cell going. So those are the only ones that are considered true blood cells. When you talk about leukocyte production, you call that leukopoiesis. Instead of hemato, you just put leuko on it. But that's how you make the white blood cells. Then if you look at the red blood cells, or the erythrocytes, you can use those terms interchangeably. These are not true true blood cells, because if you look at them, all of these little discs here, those are all your erythrocytes or your red blood cells. None of them have nuclei, so they're not considered true cells. They're going to extrude or spit out their nucleus and most of their cellular organelles just prior to when they go into circulation in the bloodstream. The production of erythrocytes, though, is called erythropoiesis, and we'll talk about that in just a minute. Then you also have the thrombocytes, and those are commonly referred to as the platelets. And those are these little things right here, and they're basically just little cell fragments of the cell known as the megakaryocyte. Mega meaning big. So that's a big cell that basically breaks into little fragments, and it forms these little platelets. Now, the platelets are important in blood clotting, and they have a very short lifespan. They only last for about 5 to 10 days in circulation. So you're constantly having to reproduce those, and that's called thrombopoiesis, when you make new platelets or new thrombocytes. But we're going to talk today mostly about erythrocytes and how everything works with those. If you look at the erythrocytes, if you take just a little millimeter of blood, you're going to have about 4 to 6 million erythrocytes, or red blood cells, in that one little milliliter of blood. They are the most numerous of all the blood cells. Their primary function is going to be oxygen transport. And so they're going to take oxygen, they're going to pick it up in the lungs, then they're going to deliver it to all the tissues, they're going to dump it out at the tissues, and then they'll go back to the lungs and they'll pick up more oxygen and they'll keep delivering it to the tissues. They also do a little bit of carbon dioxide transport, but we'll see that occurs, the carbon dioxide transport is not, most of it doesn't occur on the red blood cells. Most of the red blood cells have a lifespan of about 100 to 120 days. So you're talking maybe 3 months as a maximum life period for them. And they have a goofy shape to them. They're called a biconcave disc. Biconcave meaning two. So it's actually concave on both sides. So if you cut it, you'll see that both sides of it are concave. But then it's just like this little disc or a frisbee kind of thing. But basically, they've spit out their nucleus, they've spit out all their cellular organelles, and so they're literally just a bag of hemoglobin. That's what they have, and that's what they carry. And you say, well, okay, what are we going to do with all this hemoglobin then? Well, If the main function of red blood cells is to carry or transport oxygen to the tissues, you've got to have some way to transport it. You can't just say, oh, here's a red blood cell, let's throw some oxygen in it. You've got to have some molecule to carry it. So hemoglobin is that molecule. So it's a special protein, and it's made up of 4 polypeptide chains. There's 2 alpha chains and 2 beta chains. Excuse me. And if you remember from your 201, that secondary protein structure, you have the alpha structure and the beta structure. Don't get hung up on all that. Just know that there's 4 separate polypeptide chains that come together. So you've got one there, there's one there, and one there. And then each one of these chains also has a molecule of heme associated. So that's this little red thing here with the green dot in it. That's what we call heme. Heme is just a red pigment, and each heme molecule, though, which we've taken and blown up right here, has a central iron atom. That Fe stands for iron, if you've forgotten that. Now, it's the iron atom that actually carries the oxygen. So if you look at one molecule of hemoglobin, it can carry 4 atoms of oxygen because there's 4 heme molecules, each one having 1 iron molecule, and that's what carries the oxygen. A typical red blood cell, though, is going to have anywhere from or around 250 million hemoglobin molecules. So if each one of those can carry 4 oxygen atoms, then each erythrocyte can carry something like 1 billion atoms of oxygen. Now, I hope I've done my math right. Y'all might want to check me on that, but we'll see. But anyway, you got a lot of oxygen moving around in your body, okay? Well, as I said before, it's picked up in the lungs, and when oxygen jumps onto that heme molecule, specifically at that iron molecule, we call it oxyhemoglobin. And oxyhemoglobin is going to have a bright red color. So when you cut yourself and you bleed and you see that bright red blood coming out, that's oxyhemoglobin. It's saturated with saturated with oxygen. Then once you carry that blood to the tissues and you dump that oxygen, you're going to call the hemoglobin deoxyhemoglobin, or without hemoglobin. So you dump that off, and that gives the hemoglobin more of a dark red color. The way you get the hemoglobin onto the actual iron molecules, or excuse me, the oxygen onto the actual iron molecules of the hemoglobin is all about diffusion pressures. So if you have a great pressure somewhere and a lower pressure somewhere else, that's going to push those molecules from greater to lower concentrations. So if you look in the lungs, the partial pressure of oxygen in the lungs is somewhere of around 104 milligrams of mercury. Well, the atmospheric pressure is way greater than that. It's up almost to 160 milligrams of mercury. So that's going to push the oxygen from the atmosphere onto the hemoglobin molecule. Then when you get out into the tissues, your tissue oxygen pressure is only about 40 millimeters of mercury. And so you've got this higher pressure that's going to push it from the hemoglobin molecule out into the tissues. Don't get hung up on all that, but just know that it's kind of diffusion pressures, and that's how we move the oxygen around. All right, well, let's look at erythropoiesis, which is the actual production of the red blood cell. And as I said before, it's going to occur in bone marrow, and it all begins with the hemocytoblast. That's that original stem cell that you're going to find in the bone marrow that all blood cells are derived from. What happens is you're going to have certain proteins will come and they will act on this hemocytoblast, and they're going to cause it to differentiate into different cell lines. So we're going to find that when you want to make more red blood cells, you're going to get this stem cell or this hemocytoblast differentiating into a pro-erythroblast. Now, once it gets to that stage, it's committed to become a red blood cell. It's not going to be able to become a white blood cell after that. It's committed to that. So basically, the first thing that it's going to start doing once it gets to this pro-erythroblast stage is it's going to start producing lots of proteins, because we're making lots of hemoglobin. And remember, you've got the globin molecules are made of protein, so you've got to make lots of protein. So it's going to spend its early days of development making lots of proteins. You're going to have ribosomes that are involved in that, and then you're going to start building up all of this hemoglobin, and then you basically create this bag of hemoglobin. That's what the red blood cell is. And you don't need the nucleus anymore because you just want to be able to carry oxygen. That's the main job. So then you're going to dump the nucleus out. You're going to get rid of most of your organelles. And what you're left with is this little biconcave disc here known as an erythrocyte. And it's simply a bag of hemoglobin. That's about all it is. So it's ready to get into the circulation and start transporting our oxygen. Can't get my page undone here. Just a minute. Okay, well, what controls erythropoiesis, or the production of red blood cells? You really want this to be a precise thing, because if you make too many red blood cells, then your blood's going to start getting thick and viscous like syrup. Well, I don't know about you, but I don't want my blood thick and syrupy, because that means it's not going to be moving around the way it should, and it's going to cause problems. So when you get too many red blood cells, you get something known as polycythemia. Got it spelled right there. And that can cause all kinds of problems, whether it's blood clots or just blood not moving around easily enough and getting to the tissues and perfusing them with oxygen. Now, the opposite of that, if you're not producing enough red blood cells, you're going to get something known as anemia. And anemia is going to cause oxygen deficiencies in your tissues. So that's not a good thing either. So you want to maintain a really tight homeostatic balance between the production of red blood cells and the destruction of red blood cells. So the thing that your body, the trigger that it looks for, is tissue oxygenation. Are your tissues being oxygenated properly? If they're not getting enough oxygen, then that's going to be the stimulus that's going to cause us to make more red blood cells. And that it stimulates a protein known as erythropoietin. We'll go into that in just a minute. So any condition that's going to cause hypoxia, or when I say hypoxia, that's just when you have decreased oxygen in your tissues. Well, any condition that causes hypoxia is going to cause you to stimulate the production of more red blood cells. Possible things that would do this would be, like if you go and give blood, they usually don't take more than a pint, but you may have a little bit of, you might feel bad, and your tissues are just not getting oxygenated properly. So that's going to cause you to produce more red blood cells and replace those red blood cells that you gave when you gave the blood. If you're not making enough hemoglobin, let's say you have an iron deficiency and you're not creating proper hemoglobin, so you're not able to carry oxygen the way you should, well, that's also going to cause you to make more red blood cells, because your body's saying, we're not getting this tissue oxygenated well enough, we've got to make more red blood cells. Another thing is if you go from, like if we live basically at sea level, and let's say we go to the mountains, like I'm going snow skiing in just about a week here, and I'm really excited about that. But if I get up into the high mountains of Colorado, and I'm at an elevation of, say, 10,000 feet, I'm not going to be getting as much oxygen as I need. For every 1,000 feet that you climb in elevation, you're going to lose 3% of your oxygen. Well, you think, well, 3%, that's no big deal. Well, if you go up to, say, 12,000 feet, which is, I will probably be getting that high on some of the high peaks, you're all of a sudden, you're getting only 2/3 of the oxygen that you were getting when you were down at sea level. So my body is going to be struggling, and I'm going to be saying, I need more oxygen, so I'm going to start producing more red blood cells. So I'm going to get this erythropoiesis stimulated simply because the oxygen pressure is not as great at high altitudes. There's not as much oxygen in the air, and so I'm going to need to make more red blood cells to carry more oxygen to my tissues. All right, so let's look at erythropoiesis and how it actually occurs. It all begins in the juxtaglomerular cells of your kidney. And you're going, what? Well, it turns out that your kidneys are very much in tune with what's going on in your blood, because they perfuse, or let's just, 1.2 liters of blood actually goes through your kidneys every minute. Okay? So if you look at that, it takes about 5 minutes to circulate your entire blood through your kidneys. So your kidneys know exactly what's going on in your blood, and so they're a logical organ for this to be controlled by. So your blood's going through your kidneys, and all of a sudden your kidneys, particularly these JG cells in your kidneys, they say, man, you're not getting enough oxygen. We're detecting low oxygen levels here. So the first thing that would happen is these JG cells are going to detect low oxygenation in your blood. And once that happens, they're going to release a protein known as erythropoietic stimulating factor. Okay, so that ESF. And that's going to be released by those JG cells of the kidney, and it's going to be put into the bloodstream. Well, you've already got another protein in your blood present, and those two are going to combine and form what's known as erythropoietin. So now you've got the kidneys have stimulated this erythropoietic stimulation factor to be released. That joins with another protein in the blood, and now you make erythropoietin. Well, this erythropoietin is then going to be taken through the bloodstream to the bone marrow, where you have the erythropoiesis occurring, or this blood cell production occurring. So the erythropoietin goes to the bone marrow, and that is going to stimulate that hemocytoblast, remember that stem cell, and it's going to say, we need more red blood cells, so you start making them. And so that hemocytoblast is going to differentiate into that pro-erythroblast, and it's going to start forming red blood cells. Now, once you get these red blood cells forming, they're going to start getting thrown back into the circulation, and so now you're going to have more red blood cells and more red blood cells circulating. And as you do that, you increase your ability to carry oxygen. Well, now this increased oxygen carrying capacity says everything's fine. Our tissues are getting all the oxygen they need. So then that is going to have some negative feedback. So we're going to put negative feedback here onto our original stimulation, which was low oxygen concentration in the blood. So that's going to go back and it's going to tell these JG cells, we don't need any more erythrocytes or any more red blood cells. Everything's getting oxygenated just fine. So turn it off. So that negative feedback will turn that off and everything will be back to normal. That way you don't produce too many red blood cells. Now, this whole process is going to take about 3 to 5 days. If you have a severe blood loss, it could take weeks to replace all the red blood cells that you use. That's why they don't let you give blood every week. You've got to build back up your red blood cell production, your white blood cell production, but erythropoiesis takes about 3 to 5 days normally to get it going. So like when I go to the mountains, it's going to take me 3 to 5 days to basically get to where I need to be breathing normally and getting oxygen perfusing my tissues normally. Now that's going to be about the time that I'm going to be getting ready to leave. I'm only staying a week, so about the time I get acclimated to the mountains and that higher altitude, I'm going to have all this red blood cell production going on, and then I'm going to leave and go back to sea level and not even need it. One thing you also need to keep in mind is that your red blood cell production, or your erythropoiesis, whatever you want to call it, is also going to be dependent upon the precursors that you need to make it. So you're going to have iron, you're going to have amino acids, and you're going to have certain vitamins that are required to make red blood cells. So finally, let's say we've made all our red blood cells, and like in my case, I'm going to come back down from the mountains, and I'm going to have really too many red blood cells. I'm not going to need all that, but eventually those are going to start dying out, because remember, they don't have a nucleus, so they're going to have very finite lifespans. Usually about 100 to 120 days, which is roughly 3 months or less. And that's because we don't have that nucleus and we can't keep that cell alive. It can't produce proteins, it can't grow, it can't divide, it can't do anything. It's just a bag of hemoglobin circulating around your body and carrying oxygen. So anyway, all these older cells are always going to be filtering through the spleen. And as your cells get older and kind of worn out, the spleen has all these little tiny passageways in it called sinusoids. And these red blood cells are just kind of little bags of hemoglobin will start trying to squeeze through all these passages. And as they get kind of old and worn out, they're going to get like ripped and torn, and they can't squeeze through these passages without being damaged. So the spleen is going to naturally just kind of destroy some of the red blood cells as they age and get worn out. Well, as they break apart, remember, they're just a bag of hemoglobin, basically. We don't want to just throw away all that hemoglobin. So what happens is we're going to take that hemoglobin apart, and we're going to break it down into globins, or those proteins, which we will then take from those proteins. We'll take the amino acids, and we'll reuse them and build new proteins later on. And then we're also going to break it down into heme. And remember, heme was that red pigment that had the iron molecule in it. Well, we're going to take the iron molecule out, and we're going to save that, and we're going to reuse that. And then what's left over is going to become what we call bilirubin. Now, bilirubin is going to be taken to your liver, and it will be processed, and it will be excreted in bile. And you're going to learn later on that bile is used to break down fats and stuff like that. But anyway, it's excreted into your intestines. So you've got this bilirubin leftovers go into your bile, and then that gets excreted into your intestines. Remember, heme was a pigment. Well, once you start breaking it down and going through all these processes, it starts changing colors, that you start removing some of the things that give it color. So what makes your poop brown, your feces brown, is this excess bilirubin that's been kind of degraded down, and that's actually what turns your feces brown. So if you have white feces, what does that tell you? It says, man, we're either not making bile properly, or we've got a bile obstruction, and we're not getting bile into our intestines. Now, you're probably going to have some other symptoms that go along with that. You're not just going to wake up one day and go, golly, there's white poop in the potty. You know, it doesn't work quite like that. But it is an indication that something's wrong. If your feces start changing color, it usually indicates something. Now, you say, well, all this is occurring in the spleen, and the spleen's doing all this destruction. I know somebody who got in a wreck or whatever and had their spleen removed. How are they living? Well, you can live without a spleen. The spleen also has a lot of white blood cells in it that help you fight off infections. So basically, the two big problems you're going to have if you have your spleen removed is you're going to be prone to more infections. And then also, you're going to have more old, worn-out, kind of not really good red blood cells circulating in your body because your spleen's not tearing them up the way it should because it's not there. But you have other ways to get rid of it, and we're not going to get into all that right now. I think we're going to call it quits for this video right now, though. We'll talk some more about some diseases of the red blood cells in just a minute.